Just, I want to apologize to intervene in such a very important therapies which don't need any pharmacology or basic science. So I am pleased to show you other things, and I will be rather brief and not too precise in the science, just to tell you that, and I wanted to ask for the previous speaker if they would they would perform the same analysis of psychological trauma for person with Down syndrome, because I know that in Ukraine there is a very active association for Down syndrome through European Down Syndrome Association, which also AFRT belongs. So I just want to show you that science is very, very active, and I just tell you that there are many, many patients with Down syndrome. It's at the moment, it's still the most genetic disease, and I call it a disease. So I want to show you what's going on for science in that. The slides are not anymore. So first, we need to have precise description of the patients with very different aspects, the facial features, the speaking, the sleep, the depression, the seizures. There are a lot of new cerebral imaging. You had the previous talk about imaging from Dr. Yu, and it's done also now for people with Down syndrome. Genes and priority, animal models, cellular model. It's a very, very new area since 10 years, 15 years, and ASO techniques. So next slides. Okay. So animal models, you know that we know many of the genes from chromosome 21, and there are transgenic mice. What are transgenic mice? They are mice in which you introduce in the DNA another gene, for instance, one of the major human genes, and you have also animal models having a lot of these genes, and new methods to understand the role of each of the genes, chromosomal elimination, cellular somatic cells, oligonucleotides. I will tell you. Next slides, please. I will go fast because it's really science. Next slide. Maybe next slides. Next one, yes. So I show you the very important things as tools for study Down syndrome is that the fact there are mice on chromosome 16, 3 different chromosomes according to chromosome 21 in human. It's on chromosome 16 of the mice, 17 of the mice, and 10 of the mice. But and we can have all kinds of animal models, I will show you, but here are very important genes that we are all around the world working on. Next slide, please. But you have to know that one gene is not expressed in one area of the brain, in different area, and some genes are expressed in different type of cells of the brain, but also in different type of cells of the cardiac development, the hematopoiesis, and the ocular development. I will show you. I will go fast. Then next slide. Maybe. Okay, that's the animal models. I just want to dream a little bit. One of the major gene was found in Drosophila. Why we use all this model? You know about the mice because they are living 1 to 3 years old instead of 17 to 100 years old in human beings. But the Drosophila, these little fish live a little more than the mice, and the Drosophila live only 40 to 50 years, and the Elegans, which is a worm, lives 20 years. And all these animals can be used to study Down syndrome. Next slide, please. That's one of the animal models. It's a T66. Yeah, it's for Yann Hérault from France. And just to show you that with his model, the work of Di Domenico, it's a 20 or 25 paper. You can go to cognition, to apoptosis, to antioxidant response, to autophagy, to insulin signaling, to PERK pathway. You can do everything with mice, and even with a very important model like this one. And in this model, you can inactivate especially one specific gene and look if you have 3 copies of that gene in that complex animal, you can see what is wrong. And if you remove the gene with only 2 copies, what is better. So it's a very, very important tool. Sorry, next slide. Next slide, please. Okay, one important new area of research is chromosome 21 extension. I am not going to go very deeply on that, but it's come from a very important question years ago, 25 years ago. Why in female, in human being, we have 2 X chromosome, and in man only 1? Are we going to have more expressed gene? No. Female. No. It's because naturally the second X chromosome in female, in human being, is naturally inactivated. So doing that and explaining why the people have found a silencing chromosome with a very important techniques, and found how this can go. And the chromosome, it's a long work of some groups, and the mechanism is the 6th, the X inactivated gene, and the same mechanism, molecular mechanism, can be attained to the chromosome 21. Next slide, please. So that is the mechanism, is the CRISPR-Cas9. I don't want to go further, just to give you an idea how science is going on, and how this can rescue from chromosome 21. Of course, as I told in my abstract, in the abstract book, the main thing is most of the people in developed country ask for abortion because Down syndrome is a very bizarre and complicated disease. But if we can go further to inactivate in vivo or just after birth, because there are ethical problems, some genes, 1 or 2 important genes, it will be fantastic. And I will go further at the end of the life for Alzheimer's disease. Next slide, please. Next slide. Okay, that's researchers were able to eliminate duplicate chromosome from induced pluripotent stem cells. So another very important thing coming from basic research is to use differentiated cells, from instance from the skin. That was not for Down syndrome, but is now applied for Down syndrome and many other diseases. You have skin cells, you dedifferentiate, it's a differentiated cell, you dedifferentiate it in the lab, and you can redifferentiate in any type. It's a long work and cost a lot of money, but many labs do that. They can differentiate in different type of neurons, different type of cardiac cells, everything. So it's really combination for many different, very new approach for from basic science to apply science for some disease. And what I will show you in the next slide is the work that has been done by, next slide, please, by Jane Lawrence. She has done that for many, many years. And first she did that in undifferentiated cells, showing that they can inactivate the chromosome 21 and especially some gene. But next slide, in a very recent paper, this one, they have been shown, next one. Okay, in the previous one, they inactivate, but only in pluripotent cells. In this paper, they have shown that they can inactivate the chromosome 21 in T21 differentiated cells, especially in neural cells. And that is very important because they have shown more that these neural cells, which in which the chromosome 21 from IPS Down syndrome person have been inactivated, these cells can differentiate in neurons, and you can have good neurons which act normally. These major results open new, very important perspective for other altered 21 cells, such as cardiac, neural specific, hippocampal, hypothalamus, immune cells, and cervelet was in French, it's cerebellum, sorry. Next slide. So it's a very, very new area. There is another thing which is oligonucleotide therapy. You see that a little type of mRNA can bind to mRNA, not RNA can bind to mRNA, the altered protein, normal protein, when you know that from DNA you have RNA and then protein, so you have an altered protein, or you can also have no protein. If you find in chromosome 21, in chromosome 21, one protein which is in 3 copies, and it will be in 2 copies, you can manage the disease. Next slide. So, and this azotherapy has been already used in Duchenne muscular. Next slide. Azotherapy, this is too complicated. Azotherapy has been approved by FDA for SLA, ALS, amyotrophic lateral sclerosis, Duchenne muscular dystrophy, spinal muscular dystrophy. It's a very important tool for having, when there is a mutation on a gene, as in Duchenne muscular dystrophy, or when there is an extra copy of a gene. Next one. Next one. Past this one, it's just to see how that goes. Next one. Okay. Cardiac defect, the cardiac defect is one of the very important defects in utero, which go to spontaneous abortion in 40% of the cases of Down syndrome, of consumption of Down syndrome. And in people who live with Down syndrome after birth, cardiac defect are very important. They are different. In France, the main cardiac defect is atrioventricular canal. But in Pakistan, I wanted to show you, in Pakistan or in Turkish, it's some 2 or 3 years paper. It's the dicteroarterios septal defect. It depends the country, but the managing of this cardiac defect will be the same. Next slide. That's a story. It was looked for many, many, many years, 40 years, I think, I am in the business, to look who makes the cardiac defects, although they are different, maybe different. What is the gene responsible for that defect, which exists also in the general population, of course. And many research was done. no one was really found. And very recently, it's a paper of 225, and this gene, HMGN1, for ACV, not the other one, AVC, sorry, AVC, they'd use iPS cells from Down syndrome patients and look at the difference from others and found that the gene, which is now up on the scale of the responsible gene for Down syndrome, is this one. And through the different techniques that I showed you before, you can see, next slide, that you can make, of course, you found that expression of these genes in trisomy 21 mimic AVCM, and that if you reduce in animal models, for instance, this one is a DP1 type of, it's done in England, they can rescue the cardiac defect. Though it seems that this gene precisely is responsible for the cardiac defect. So if you can manage in utero or very early after cardiac formation in utero, which is around 2-3 months, so you can manage, maybe, I don't, but there are ethical problems for that. So next slide. So creation of iPS cells from Down syndrome, you can see that the cord could be differentiated in 3 germ lines. The iPS can be used for studying cellular and developmental etiology of congenital heart defect induced by 3 copies of these genes in Down syndrome iPS cells. Next slide. Okay, that's the same for 2 patients from the, so combining iPS cells in the inactivation and silencing one gene, you can have new perspective for Down syndrome. Next slide. Important problem, yes, mice are good, but it's important to know the defective patients very early. Next slide. So you have a lot of paper. This is a slide coming from a person who is Nelly Bitelou, who is in Switzerland and who has made a trial with a gonadotropin releasing hormone. But I want to show you just how in the cerebellum, sorry for the French, the total volume intra-brain, the volume of the white matter and the volume of the gray matter is different between control and Down syndrome, between control and Down syndrome. And it's the same for the this is for the total, and this is in cerebellum. So looking at therapy means that you assay very important things, characteristic into the brain to look if it's effective for, and we have seen from Dr. Liu that intracranial therapy can be, acupuncture could be very good. Next slide. And a lot of things have been recently done with connectivity in Down syndrome, the white matter, and also something very interesting is now generative IA can give models giving biological signification to some neuroanatomic characteristic present. So it's a generative artificial intelligence who can give new correlation between what you see in brain and what are the phenotypes of the person. Next slide. Now we are going to the Alzheimer's disease. Maybe you know that APP, the gene which gives to the senile plaques, is on chromosome 21, and it was already seen by Professor Alzheimer at that time. So we knew since a long time that APP was on chromosome 21. So that's the comparison between Alzheimer's disease and Down syndrome. And so there are executive function, memory, language, attention, and learning, which are defective, and a lot of things. And so Down syndrome, I think in the next slide, you will see there are a lot of slides for Down syndrome. You can go fast to pass the slides. Next slide. Okay, you see maybe the main feature is neural death, neurofibular tangles, and microtubule disintegration, and you have plaques and neurofibular tangles, which is the same in Down syndrome and in general Alzheimer's disease. Next slide. Okay, that's the same. You see the microtubules are effective, the amyloid way, and you have the different plaque, diffuse plaque. So I give you a course on the Alzheimer's disease in the same time, core plaques and disease, dense, diffuse and dense core plaques and compact plaques. Different states. Next slide. Okay. Diminution of the brain, cognitive deficit, spatiotemporal. I saw you. Poor smelling. It's a fact that there is a poor smelling in person with Down syndrome and also in Alzheimer person without Down syndrome. Sleep disturbancy and many other things. Next slide. Go ahead. Go ahead. Next slide, please. Okay, that's the early Alzheimer biochemical signs are present very early in young adults, and the signs of dementia are the same as in Down syndrome. So Down syndrome is one of the genetic model for Alzheimer's disease. The prevalence of dementia increased with age to near 75% at 65 years, but even some people with Down syndrome will never develop Alzheimer's disease without any molecular deficit in APP. There are still 3 types of APP genes, although it has been reported that some very rare person with Down syndrome have only 2 APP genes instead of 3. Next slide. Okay, some of during early childhood, early in the course, pharmacological intervention to decrease this deficit in DS may help to understand better early deficit in the general population with Alzheimer's disease. Next slide. Okay, that's an interesting slide. That's the general population load. The mean in blue is the mean time of it to appear, Down Alzheimer's disease, and in red the age of at death. You see in Alzheimer's disease, in genetic families, in families which have a genetic mutation, either on the gene PSV1 or PS2 or in APP, that the 3 very rare families, you see how the disease starts very early, 43, 58, 47 for mutation in APP, and there are very rare families with a duplication of the gene of APP, so they have 3 APP genes, but only these 3 APP genes, although in Down syndrome there are 3 APP genes and other 3 copies of chromosome 21 genes, but you see the application of APP give a very early state of the disease with an early death, early age of death, 60, and it's almost the same in Down syndrome. It's a very interesting paper, which is rather old. It's from 2021. Next slide. I will go fast. Okay, that's without Down syndrome, healthy Down without Down syndrome, Alzheimer's disease with Down syndrome, healthy Down syndrome. We see early diffuse plaque, APP plaques, very early, and you see Alzheimer's disease with Down syndrome. So you have magnetic APP load. APP means the peptides which are derived from the APP gene. It's small peptides of 41, 42 amino acids. And neurofibrillary tangles are the late sound. So, and at the very low, you have the gene RANK1 and the gene DIRK1A. Next one. Okay, that's the marker that you can have in the general population. I don't want to go too fast. Some are the deficits, some are increased. It depends, because I will be too long if I go too in deeply. Next one. Next one. Okay, prediction. So that's a very recent paper with 250, almost 60 adults, age 25, follow-up for 16 months in UK, and you have the markers, plasma PTO, and GFAP. So it's very important to have markers which can help you to find the disease before it appears. So that's one of the next one. Risk factor, I show you, APP, DIRK1, there are a lot. Next one, many genes, so a lot of study, APP, DIRK1A, RANK1, S1 beta, SOD1, and other gene, CBS. Next one. If you find, and this is a paper, so a paper of 25, almost these genes, 11 genes in addition to APP, directly or indirectly involved in mechanisms related to pathophysiology of Alzheimer's disease in Down syndrome. Next one. Okay, that's the new pharmacology. One of the genes which is very important is DIRK1A, and I will show you that DIRK1A has been studied in very different type of complex animal models, mouse animal models, or even rat, and can be used as a genes involved in brain development or in Alzheimer's disease. Next one. DIRK1A, and also DIRK1A is not only involved in, as I told you in the very, very first diapositive, is that it depends. One gene is said to be involved in brain development, and now we found that DIRK1A is involved in the blood development and immune response. So if you decrease the expression of that gene, you may improve many different aspects than the brain aspect that you were initially focused on. Development. Next one, it's about DIRK1A. Next one. It's involved in inflammatory responses, and it's very important because in Alzheimer's disease, there is inflammatory aspects in the general population and also in Down syndrome. It's also, DIRK1 is also involved, but not directly, through partners in leukemia, which is another part of the story of Down syndrome. Next one. It's involved, next one, it's involved also in viral infection. And okay, maybe that's explain why there were so many problems with when there was in the infection with COVID in people with Down syndrome, with a lot of deaths at the beginning. Next one. Okay, that's type. So many labs are working on inhibition of DYRK1A, and you understand that according to type of inhibitor, you can go to the immune system, to the inflammatory system, to the Alzheimer's system. Next one. That's a lot of implication. The first one was ECGG. In Down syndrome, it was thought that green tea which is an inhibitor of DYRK1A in early development, could be a good drug, but it happened that it was not really efficient. But with remodeling NCG like not competitor, and there is a new paper which maybe which give hope to a very different, a very close type of peptide, but not exactly the same. Next one. Next one. There is another gene which is very important for Alzheimer's disease. It's a calcineurin 1, which is involved in Down syndrome also, and it can be a good competitor. Next one. Down syndrome, that's the same as the one. So you have the same I saw you before, and with interrogative for calcineurin. Next one. There is a trial. It has been finally synaptic therapy provides an opportunity to prevent AD progression before it begins in Down syndrome and in Alzheimer's disease in the general population. So you see, looking at Alzheimer's disease in Down syndrome can help Alzheimer's disease in the general population. Next one. Ongoing trial. There are a lot of ongoing trial, just to show you that we can intervene in the beginning of the life of person with Down syndrome, or even in utero with ethical problem aspects, and in the old age. Next one. Okay, that's one copy looking at Down syndrome with three copy, one copy, and you manage. And just looking at DYRK1A, it was found there is a new disease which is called DYRK1A disease, which is the person who have mutation on DYRK1A and who have some mental retardation. So looking at one gene, which is on chromosome 21, it happens that you have new sites in the person with other disease, which is not Down syndrome, which is DYRK1A disease. Next one. Okay, that's the DYRK1A syndrome. Too little DYRK1A or mutation of DYRK1A give you in embryo somebody who would be, and they can survive, and you see all the models, the mouse, the chick, the xenobus, the xenopus, for both Down syndrome and DYRK1A, which is just found because DYRK1A was used in basic science, and it was seen that a few person with DYRK1A mutation have all these diseases, and you can manage in different animal models. Next one. I think I am almost done. Main conclusion, many animal models, the use of IPS, the DYRK1A operon, other gene like CBS. Thus, we are really at a new area for people with Down syndrome, for even much better life of person with Down syndrome as today. But I want to say that although the expectancy life was 30 years old in the 30s, it's now 65 years. That's why it's very important to look at both aspects of the life. At the end, the cardiac problems, and at the end, the Alzheimer's disease, and in between, the society should help to have a better insertion. And I want to tell you that AFRT has settled in 2005 the idea of having a Down syndrome day in the world, and it's now made in many, many countries, and there was last 21st March, something in New York, something in Geneva, something in Brussels, and something in France, and in many, many different countries, including, I am sure, Ukraine.